Hello all. The following is a presentation on galactose oxidase. This metalloprotein was studied during bioinorganic chemistry, and this presentation was compiled as a portion of the class. This is presented by Jason Mills, a chemistry major at Georgia Gwinnett College in the fall of 2020.
Hello everyone. Seen in figure 1 is galactose oxidase with its central copper atom. Galactose oxidase is a copper-containing metalloenzyme. It contains 639 amino acid residues and is coordinated by 5 ligands. Traditionally, chemistry students are taught in organic courses to use the chemical pyridinium chlorochromate, PCC, to facilitate the oxidation of a primary alcohol to its aldehyde form. Upon dissection of the name PCC, one can note the presence of chromium, a known toxic substance. As an alternative to this toxic, yet effective, chemical pathway, one can look to the utilization of GO when pursuing greener chemistry techniques. This enzyme GO can be very useful in the field of synthetic chemistry, where the oxidation of alcohols is a common reaction. Most compounds, like PCC or permanganate, used to catalyze this oxidation are expensive and/or toxic, making biocatalysts more desirable. The high regioselectivity and stereoselectivity of this enzyme make it a very favorable catalyst. Galactose oxidase was believed to be discovered in the fungus Polyporus circinatus in 1959 by John Cooper, Wilfred Smith, Metri Bessela, and Heider Medina. It was in a cell culture of this fungus that it was first isolated. However, a few years later, it was found that the fungus had been misidentified and was actually Dactylium dendritis. Displayed on the left in figure 2 is the Gyromycete fungi Hypomyces rosellis. This is also known as Dactylium dendrites, or commonly as cobweb mold. Note the rose color and web-like texture of this mycelium. These spread across the fungus host, especially Aphilophorales, or the wood-rotting fungi. The molds start out white and later develop their pink color and pimple-like perithecium, or sporing bodies. In figure 3 is a georeferenced map denoting 644 logged locations where this fungus has been found. In 1965, Bloomberg and his team further studied this material and found that the copper central atom was largely in a 2+ state through examination of the electron spin resonance spectra. In figure 4, we see the active site of galactose oxidase. One can note that it has 5 ligands, and this gives it a distorted square pyramidal structure. These consist of a tyrosine in the axial position and 4 equatorial ligands, 2 histidines, another tyrosine, and a 4th labile bond with the solvent molecule make up the planar ligands. In this case, the solvent molecule is a primary alcohol bonded to the hydroxyl group. Note the radical in the aromatic ring of tier 272. This is stabilized through delocalization and was a topic for debate. The protein radical interacts antiferromagnetically with the copper ion, resulting in an EPR silent species. This is an unusual result when dealing with copper enzymes, as typically there is a strong defining peak signaling the radical's presence. However, due to the antiferromagnetically coupled electrons of copper in the tier 272 delocalized residue in figure 4, this results in a silent species that would not give the expected spectra, falsely implying that the radical chemistry doesn't take place. This mechanism will be discussed next. Once again, as detailed in scheme 1, this enzyme is used for the oxidation of primary alcohols to their corresponding aldehyde form. As just mentioned, GO is useful in the redox conversion of primary alcohols to aldehydes. This is done in what is known as the proton-coupled electron transfer mechanism. This consists of 3 concerted steps and 1 regenerative step. Looking again at the active site, we see the starting 5 ligands signaling the distorted square pyramidal structure. This undergoes its first step, or proton transfer, from the primary alcohol, and tier 495 breaks off. This now is a 4-ligand structure, giving it a square planar coordination. This is characteristic of a low spin state. Note the location of the radical in 272. This will be transferred to the alcohol at the alpha position from the ether in the hydrogen atom transfer step. We still see 4 ligands in this structure, and this will undergo an electron transfer step, ejecting the alcohol as an aldehyde after oxidation. 3 ligands are now observed, and this is a trigonal planar structure. Copper has been reduced, becoming copper 1. This is an increase from 9 d electrons to 10. It can be noted also that the magnetic moment has undergone a change from 1.732 amperes per meter squared to 1.414 amperes per meter squared. Tier 495 will reform its bond, and a new primary alcohol will be introduced by the reduction of dioxygen, ejecting hydrogen peroxide. This restarts the cycle. This protein, GO, has many uses and is found commonly in the field of biotech, as well as food and pharma processing. In conclusion, there are many applications for this enzyme over a wide variety of chemical industries, including biotech. Galactose oxidase is used in the synthesis for food and pharmaceuticals. It is a competitive and cost-effective catalyst compared to chemical conversion for the manufacturing of fine chemicals for pharmaceutical purposes or in the food industry. For example, GO was used for conversion of sugars like D-galactose to food-grade cross-linking agents. The galactoaldehyde products produced from the oxidation of galactose have also been useful in the production of materials such as hydrogels and aerogels. Further, GO has found use in biosensors for the detection of galactose in biological fluids. Galactose oxidase can even be used to modify carbohydrates in cell walls, making it useful in cell labeling and histochemical staining. Thank you for your time.